Every atom in your body right now is older than the solar system. The carbon in your left hand was forged in the core of a star that exploded 4.6 billion years before the Earth existed. The iron carrying oxygen through your blood was synthesized in a supernova whose light has long since faded from any sky. You are, in the most literal sense, permitted by physics, made of dead stars. And right now, all of those atoms, roughly 7 times 10 to the 27th power of them, a number so large it has no common analogy, are arranged in a specific pattern. That pattern is you. The question is, what happens to it when you stop? The first thing to understand is that death, from the perspective of physics, is not an event. It is a transition. The atoms do not go anywhere in the first 60 seconds. Not a single one of the 7 octillion atoms in your body leaves. What changes is not the content. What changes is the control. Your body runs on a hierarchy of electrical signals. At the top of that hierarchy sits a structure in your brainstem called the reticular activating system, a dense mesh of neurons roughly 10 centimeters long that functions as the master clock and coordinator of every process in your body. It keeps your breathing rhythmic, your heart rate calibrated, your gut motility timed, your hormone release pulsed at the correct intervals. When blood flow to the reticular activating system stops, which happens within 10 to 15 seconds of cardiac arrest, that coordination collapses, not gradually, precipitously. In those first 15 seconds, the neurons in your prefrontal cortex, which require more oxygen per gram than almost any other tissue in the body, begin shutting down. The prefrontal cortex consumes approximately 20% of your total oxygen supply, despite representing only 2% of your body's mass. When the pump stops, that demand cannot be met. Glucose delivery to the synapses fails. The sodium-potassium ATPase pumps embedded in every neuronal membrane, the pumps that maintain the electrical potential difference of roughly minus 70 millivolts that allows neurons to fire, lose their energy source and begin to fail. The membrane potential collapses. You lose consciousness. But here is what the atoms are doing during those 15 seconds, because this is not the same story. The atoms are still bonded. Every covalent bond, every shared electron pair holding carbon to hydrogen, oxygen to carbon, nitrogen to carbon is intact. The ATP molecule in a neuron that just fired and hasn't been replenished yet is still ATP. The water molecules in your cerebrospinal fluid are still water. The hemoglobin in the red blood cells, still in your arteries carrying oxygen that will never be delivered, is structurally identical to the hemoglobin it was 30 seconds ago. Chemically, at the atomic level, the first 15 seconds are nearly silent. The deeper silence is biological. The systems that have been maintaining order, that have been fighting thermodynamic entropy every second of your life, have stopped receiving instructions. Here is the scale of that maintenance work, because it is extraordinary. Every second you are alive, approximately 10 million of your red blood cells are destroyed and 10 million new ones are produced. Your intestinal lining replaces itself completely every 3 to 5 days through a process requiring constant cell division, constant protein synthesis, constant energy expenditure. Your liver runs 500 simultaneous metabolic processes at any given moment, detoxifying ammonia, synthesizing cholesterol, managing glycogen stores, producing clotting factors. Your immune system patrols your tissues at all times, killing bacteria that have crossed your skin barrier, clearing cellular debris, running surveillance programs that require active signaling and active response. All of this costs energy. All of this costs order, and all of it is what thermodynamics has been trying to undo every single second of your life. Death is not the arrival of disorder. Death is the removal of the systems that have been resisting disorder. In the seconds between 15 and 60, those resistance systems begin to fail in sequence. The sequence is determined by oxygen dependency. The brain goes first because neurons are catastrophically oxygen sensitive. A neuron kept at 37 degrees Celsius with no oxygen supply begins to experience irreversible damage within 4 to 6 minutes, a window so narrow that it defines the entire architecture of cardiac resuscitation medicine. But the first signs of failure begin within seconds, not minutes. At around 20 seconds, The neurons in the hippocampus, the structure particularly dense with NMDA glutamate receptors, begin releasing glutamate in uncontrolled bursts. This process is called excitotoxicity, and it was characterized in detail by John Olney at Washington University School of Medicine in the 1970s. Neurons, deprived of the energy to maintain their resting potential, fire uncontrollably and release glutamate, faster than the surrounding cells can clear it. The glutamate binds to NMDA receptors on neighboring neurons and triggers calcium influx. Calcium, at physiological concentrations, is a second messenger. It carries information. At the concentrations produced by excitotoxicity, it is a catalyst for cellular self-destruction. The atoms here are doing something specific. The calcium ions, each one a single calcium atom that has lost 2 electrons and carries a charge of +2, are flowing through open NMDA receptor channels down their electrochemical gradient. Each channel allows approximately 10 to 100 million ions through per second when fully open. The movement of those ions is not metaphor, it is not abstraction, it is matter moving through protein pores in a lipid bilayer, because a charge imbalance exists and physics permits the flow. At 30 seconds, the heart muscle, the myocardium, is also in trouble, though the timeline is longer than the brain because cardiac muscle can tolerate anoxia for slightly longer than neurons. The cardiomyocytes, which have been contracting 60 to 100 times per minute for every minute of your life, require continuous ATP production to maintain function. Each contraction of a cardiomyocyte costs roughly 2 ATP molecules per myosin cross-bridge per cycle. The heart at rest burns approximately 6 kilograms of ATP per day, far more than its own mass, recycling the molecule continuously through oxidative phosphorylation. When oxygen delivery stops, oxidative phosphorylation fails within seconds. The cells switch to anaerobic glycolysis, which produces ATP at 1/18th the rate of aerobic metabolism. It is not enough. The cells begin to accumulate lactate. Intracellular pH drops. This is where the atoms become visible again, because pH is not abstract. pH is the concentration of hydrogen ions, individual protons, the smallest possible charged particle in solution. When the pH of a cardiomyocyte drops from its normal 7.2 towards 6.5, what is happening is that protons are accumulating. Each one is a hydrogen atom that has lost its electron. They are binding to proteins, changing the shape of enzymes, disrupting the molecular geometry that makes biochemistry function. The sodium-hydrogen exchanger in the cell membrane, a protein that normally pumps protons out of the cell in exchange for sodium, reverses direction under acidic load. Sodium floods into the cell, water follows osmotically, the cell begins to swell. Here is what is extraordinary about this. The cell swelling is occurring because of individual atoms following individual physical laws. Water molecules, each one 2 hydrogen atoms bonded to 1 oxygen, a structure with a partial negative charge on the oxygen and partial positive charges on the hydrogens, are crossing the cell membrane through aquaporin channel proteins because the solute concentration inside the cell has increased. They are not being summoned, they are not responding to a signal, they are moving down a concentration gradient the way water always moves down a concentration gradient. Physics is taking over from biology. And here is the distinction that matters. Biology has always been physics. The difference between a living cell and a dead cell is not that different physical laws apply, it is that a living cell is a machine that uses physical laws to maintain a state far from thermodynamic equilibrium, and a dead cell has simply lost the energy input required to run that machine. The atoms in both cells are following identical rules. One set of atoms is organized by a continuously funded maintenance program. The other is simply following the gradient. At 45 seconds, the process spreading through the brain reaches what neuroscientists call a cortical spreading depression, a wave of electrochemical silence that propagates across the cortex at roughly 3 to 5 millimeters per minute. This phenomenon was first described by the Brazilian physiologist Aristides Leão in 1944 working at Harvard, who observed it in rabbit cortex during experimental ischemia. Liao noticed that a wave of near-complete neural silence moved across the cortical surface following an initial burst of activity, leaving tissue that appeared intact but electrically inert in its wake. The wave is caused by the mass failure of the sodium-potassium ATPase pumps, which produces a sudden, massive shift in ion concentrations across neuronal membranes throughout the affected region. Sodium and calcium rush in, potassium rushes out, the cells depolarize completely, firing one last mass discharge, and then go silent. The atoms in that discharge are doing something that has never been fully mapped in human subjects until recently.